Hey guys, it's Liz. I'm just here today to give you a quick rundown of what we did last week in lab, to go over the Lab 8 food habits analysis lab with you guys. If you've talked to your lab mates by now, you know that the assignment is pretty short, and I basically just allowed them to complete it in class together, and then they submitted it to me via email when they were done. And so, because I was kind of set back by COVID this past week and wasn't able to record some lectures for you guys, I'm going to go ahead and extend the deadline for this for another week for everybody. And if you follow along for the next 25 minutes here for this video, you should be able to finish this lab pretty easily. We'll get through all of it together in the next few minutes here. Let's see, and with that, I'll go ahead and open up the Lab 8 document here. Share my screen. Okay, here we go. So, so as you can see here, we've got this food habit analysis document open. And so diet analysis is a really important tool for fisheries biologists. It can tell you a lot about a population, getting a sense for what prey fish are utilizing in different impoundments can give you an indication of whether or not fish are stunted, whether or not there's sufficient prey for them, whether or not there's an invasive species that's affecting their diets. There are a lot of different strengths or merits to doing a diet study. I myself am doing a massive diet study for my dissertation, and we're trying to look at gizzard shad and evaluate whether or not gizzard shad are an important forage fish for largemouth bass across small Kansas impoundments. And so to do that, there are multiple different ways to analyze diet, and we're going to look at some of those today. So to start with, we're going to talk about the classical procedure for doing diet analysis. So the first step is to collect your fish via electrofishing, seining, what have you. And then you need to preserve the fish. You can freeze them, or you can fix them in formaldehyde or ethanol, any preservative, or you can do either whole fish or just the guts only. And you always want to make sure that you're carefully labeling them with your size, your date, your time, the location that you collected them, and the means of capture, whether that was electrofishing, seining, what have you, trap netting. So then the fourth step is to remove the stomach and identify the stomach contents. And so usually with fish, the stomachs are small enough that you need a microscope to be able to do some of the more fine-scaled analysis. And then we do the analysis contents, we do quantification, volume estimation, we can weigh and measure the different contents to get different estimates. And so here's just a diagram of three generalized shapes of fish stomachs. And so up here in number A, that would be a stomach of a piscivore or an omnivorous fish. This is the generalized shape. So here we've got this tube that's the stomach itself. All of these squiggly lines are the pyloric ceca branching off at the base of the stomach and then feeding in. This would be marking the very end of the stomach itself, and then where the pyloric ceca ends and beyond would be the intestine leading to the anus. And then we have, so this would be like a largemouth bass or something of that sort. Whereas in B here, we have another piscivore. So this would be like a white crappie or a white bass or striped bass type stomach. And so you see we've got an esophagus, a really short, stubby stomach, the pyloric valve, and then the pyloric ceca here, and the intestines trailing off. And so you guys will notice that for piscivorous fishes, the stomachs tend to be smaller. And that relates exactly to like cell ratio, cell volume theory, right? In terms of if you have greater surface area, it allows for greater absorption, right? With greater diffusion across cell membranes. And so if you have a longer gut, if you have more intestines, longer stomachs with more surface area, you're going to have more time and it's going to be easier for you to absorb materials passing through your gut. And so for a piscivore that's feeding on high protein quality food like fish, they don't need as much surface area to break down some of the material. Whereas, for example, a detritivorous or a herbivorous fish like a gizzard shad or a golden shiner, or any fish that's eating primarily plants and detritus, so like a common carp, you're going to have a really elongated gut to create that surface area. There'll be a lot of twisting and coiling, and really bunching up all of the gut and the intestines together to create just more time and more length across which to absorb some of the materials as they're breaking down, because it's harder for stomachs to break down like any kind of woody vegetation or plants that you have to break down cellulose, you have to break down lignin, and all of those take more time for the digestive enzymes to be able to break those things down. So it all just depends on what kind of macromolecules the fish are consuming. And so those are the three generalized types of fish stomachs. And so there's a lot of different analysis techniques that we can use. The first one, and one of the most common ones, would be numerical analysis. And so for numerical analysis, you're just counting the number of each food type in the stomach. So you classify items by type and then count them. And then you calculate the percentage by number in each stomach and in the whole collection of stomachs, so multiple stomachs across your sample. And then you would assume that the food most ingested is, the most nutritionally important, but that might not be the case, right? So just because something makes up the bulk of a fish diet doesn't necessarily mean that it's the most important source of energy for them, right? A fish could be occasionally consuming really high energy prey resources, like say a bass eats a bluegill, and that's like really concentrated protein, right, and high caloric content. Or it could be expending less energy in like single events, but having to spend more time foraging for lower value things like a little water boatman, like insects, things like that. And so, yeah, we have to keep in mind that the food ingested the most might not actually be like the most nutritionally important. It might just be what's there. The next analysis technique is where we calculate the frequency of occurrence. And so this is like one technique that I use a lot and we see a lot within the literature. So, and for frequency of occurrence, you're recording the number of fish in which food item occurred, and you express it as a percent of all of the fish in your sample. The only problem with this is that it doesn't provide any information on the bulk, like how much of each food item was present in each stomach, or the numbers consumed. And so hopefully that's kind of clear what I'm talking about. The bulk would be just like the volume, the mass and the areal volume taken up by the food items consumed, whereas the number representation would be like, let's say there were 7 whole mayflies we were able to differentiate in one stomach, and in another stomach they had 30 mayflies of different sizes. But those two stomachs also have different volumes of mayflies in their stomach. So the problem with frequency of occurrence, like I said, is that it doesn't give us information on the bulk or the numbers consumed. And it also can overemphasize the importance of really persistent items. And so it overemphasizes things that show up regularly. And so that's a source of bias that we need to acknowledge when we use this as our metric. The next two analysis techniques kind of go hand in hand. So the first is the gravimetric analysis. And so you're determining the weight of each food type in the stomach. And so you express that as a percent of the total weight of everything in the stomach, as well as a percent of the total weight in the entire sample. So like the percent of total weight in just the single fish compared to the total weight of all of the food items of all of the fish that you sampled from a particular impoundment. The problem with gravimetric analysis, again, is that there's distortion of items, the distortion by items of really large weight. So if you have just like a few items, like a crayfish or something, and then the rest of your samples don't have crayfish, that crayfish is going to be a lot heavier and it's going to skew your sample, right? And then we have volumetric analysis, similarly, where you're measuring the volume of each food type in each stomach. And this is often done via displacement. And so traditionally, fisheries managers would like take the sample of diet from an individual stomach and they would put it in a tube with a known volume of water. And then once they added it, they would measure the change in volume in a little graduated cylinder, and then that change in volume in the cylinder was the volumetric measurement for that sample. And so you can calculate it if you calculate the total volume of a fish stomach. You can separate out each of the prey items by type and calculate the percentage of each type as a percentage of the entire volume. Unfortunately, volumetric analysis, again, can be distorted by those slow digesting large volume items. So the really big, bulky stuff like crayfish, that's going to distort it. All right, so the next method is the points method. We won't spend a lot of time talking about this, and we're not going to use this semester. It's not used that often, but it's an approximate volumetric method that's useful in the field, but it's weak in that points are subjectively assigned. And so an example of this is where you dump, like basically you could be doing gastric lavage, which is where you pump air into a fish's stomach using a tube, and it causes the fish to vomit up whatever they ate that morning. And so what you can do is if you don't have a lot of time or preservative to be able to take those samples back to the lab, you can perform gastric lavage, and then on that tray, you could have little points or grids or squares to kind of give you a representation of areal volume. And then however many points or grids that a particular volume or a particular sample of stomach matter covers, you can represent that as like X number of points for one fish versus X number of points crayfish for a different fish of the same species. And so it's a really loose way to get at volume, because it's like areal volume. And so this would be, I wish you guys could have been in lab the other day, because I had back at the microscopes in the back of the lab when we were doing some of this with our back samples from the tallgrass preserve. For each microscope, I have these little 1 millimeter by 1 millimeter grids, and there are like hundreds of them, right, on a sheet we have printed out. And then you can do something like the points method, where you calculate, you can count how many of those 1 millimeter by 1 millimeter grids a particular prey item covers, and that can be used as an approximation of like a volume or a mass, especially if you do that multiple times, like many times, with a particular type of prey item. You can create, if you, for some of those samples, actually take the masses of the prey items, then you can create a relationship, right? Like you can calculate an R-squared and plot that relationship between number of squares or points and the actual mass to get a relationship between those. And you can use that as a rough estimate. It's not perfect, right? I do this sometimes, but it's definitely like a hard and fast way to estimate fish diet. All right, next up, we're going to talk about hybrid indices. And so these combine different metrics like the percent number, your frequency of occurrence, and the percent weight. The benefits of a hybrid indices would be it reduces your bias associated with the other methods, where, you know, like a large volume item throws things off, that kind of thing. But there's also the bias of summing and multiplying proportions. And so you have to be really careful when you utilize a hybrid index, because sometimes when we sum and multiply a bunch of proportions all at once, it can result in a number that doesn't really mean anything, like we can't interpret it. And as fisheries managers, if we get an index that like we can't use and can't interpret and share with the public, then what does it matter, right? Why did we even do it in the first place? And so finally, for number 7, the best practice and the best results attained are usually from a combination of all of these methods. And so the most commonly used include the numerical one, the frequency of occurrence method, and then a volumetric or a gravimetric method. And so we're going to do something kind of like that for lab today. So you can always supplement food habits studies with studying the kinds and amounts of food available to fish, the frequency of feeding. You can look at food preferences, like what are fish selecting for. Sorry guys, I got a street sweeper outside. Sorry, the pesting. I'm so sorry. Okay, much better. Sorry about that. I know why the city of Manhattan insists on continuing to, sweep the leaves out of the street, but it keeps happening. So anyhow, some other things you can supplement food habits with include daily food requirements. So if you can calculate the caloric needs of an individual or the average caloric needs of fish of different sizes for a species, then you can look at, are they consuming whatever it takes to meet their minimum daily caloric needs. And maybe if they don't in a particular impoundment, you can collect more data to figure out, well, what is it they're eating and why are they not eating higher caloric food? Is it just not there, or are they just not selecting for it? You can also do analysis on the nutritional value of foods. You can study the efficiency of food conversion and then digestion rates. So my lab mate, Sky Hedden, has worked with my advisor Keith for a long time on this really cool project looking at channel catfish digestive rates at different temperatures out in the San Juan and Gila River basins. And so basically Sky would go out and you would take catfish and keep them in little holding tanks, basically, and he would come back he would allow them all to feed at the same time, and then some of them would be in one temperature group, and some of them would be in another temperature group, because he did this both in like spring, summer, fall, winter. But he would basically take all of these fish after they had been fed and keep them in these buckets, and he would uh, take a subsample of each of them from that initial sample. And some of them he would do gastric lavage on at 4 a.m. If he fed them at midnight, he would do one sample at 4 a.m., one sample at 8 a.m., another one at like maybe 2 p.m., one of them at 10 p.m. at night, and then again at midnight. And then he did that for multiple days, because sometimes at the colder temperatures in wintertime, catfish would take like 3 days to digest a fish that they had swallowed. And so he was able to calculate digestive rates for catfish of various life stages and at different temperature regimes. And that kind of information is invaluable if you're doing a diet study to figure out like how fast are these fish digesting things? What are their caloric needs? How much are they digesting? Because the digestive rate can give you a rough estimate of the daily food requirements over time. And it's important to remember that food habits can also vary with habitat. They can vary with season. So some species will eat one thing early in the spring, and then they'll eat another thing when a particular insect hatches, and then they'll eat something later in the fall. And so some species undergo ontogenetic diet shifts throughout the course of a single season. And when I use the word ontogenetic, I think I used it in class before, but that just means something shifts within a fish over the course of life stages. So going from a larvae, right, a fry, a fingerling, to an adult. At those different ontogenetic stages, there are different food requirements and therefore different food habits and feeding preferences. Food habits can also vary with temperature, kind of like I mentioned with the catfish, depending on the temperatures. Temperatures affect fish behavior, right? Because fish are ectotherms, and so they're unable to regulate their body temperature, and basically the temperature of the water has a direct impact on their metabolism. And then the fish size plays a role. So bigger fish tend to have bigger caloric requirements, right? You've got to have more calories to maintain more tissue. The time of day plays a role, the relative intensity of competition, both within species and between species competition for the same resource. And then just food availability, what's there that they can eat. That all influences the habits of fish and what they're eating. All right, so now we're going to get started calculating some of these different feeding indices. So the first one that we're going to talk about is the IRI, the Index of Relative Importance. And so the IRI is basically a combination of three measures of prey importance. So the frequency of prey, the number of prey consumed, and the volume. And it allows for a more representative feeding habit data summary while still allowing for each measure to be evaluated individually. And so you calculate IRI as percent N plus percent volume, so the percent number plus percent volume times the frequency. And so here's an example IRI table. So we've got 7 different prey species over here, and then we've got the number percentage of each of the different prey. And then we've got the volume consumed as a percentage of the total. And so this would be the most important prey by volume, and then like the second one going down, but actually they're sorted by number here, and then it follows by volume and frequency. And then we've got the calculations for IRI over here where they're taking this value for the percent number. So 30% of all of the different items in these stomachs was this first species, 18% was the second species. So let's say this is a green sunfish, this would be a bluegill, this could be a dollar sunfish or something like that going down. And then we've got so this percent number plus percent volume times the frequency will get us. Usually the first ones for the sample can be over 1,000. It's okay to have IRI values over 1,000. Okay, let's see here. So again, it's combining some of those data. And so here's another example of the index that combines data. And so that's the percent similarity index, the PSI. So we're doing the IRI, and now we're talking about the PSI. And so the PSI is combining qualitative, so presence absence data, like a 1 or a 0, right? If it's not present, you give it a 0. If it's present, you give it a 1. So those binary variables. And then it's also quantitative abundance information. So like 0 here, 16 or 3 there, right? And so both of those types of information on the feeding habits. And you can do it between individuals, you can do it between species, or you can do it between age classes of the same species. One caveat of this particular index is that the PSI can be influenced by rare diet items. So you calculate the PSI as the percent similarity of species X and Y is equal to the sum of the minimum value for species I or species 1J, and then the minimum value of species 2. And so it'd be best to just kind of go through the examples for these. So here we have the counts, like how many hits we had for species X. So let's say this is a bass, and let's say this is a channel catfish. So species X is our bass predator, species Y is our channel catfish predator. So 4 bass out of the sample had, let's say bluegill is our first prey species. 5 bass had the second species, so maybe green sunfish. And then we had a couple of bass that didn't have either of these two species, let's say like an orange spotted or a dollar sunfish. It's 7 bass that had prey species number 5 and 9 that had number 7. And so the sum of that would be 26. Same thing going down the catfish. We had 1 catfish swallow a bluegill, 1 catfish swallowed a dollar sunfish, and so on, for a sum of 10. And then, so this will be the percentage of each of these. So, so for species Y, we had 16% of all of the prey was species 1, 20% of all of the prey items was species 2, 28% of all of the items was species 5, and 36% of the items with species 9, and we get those by reflecting like 4 out of 24, out of 26, like 5 out of 26, and so on, going down the line through that. And so then, and so that's for, oh, excuse me, like 1 out of 10 versus 1 out of 20, or 1 out of 10, 2 out of 10, that kind of thing, gives you those values. For species X here, oh my gosh, here's that again. We do the same thing. So it's like a representation of the totals, or the counts, and we go from there. And so you're trying to go for the minimum value for each of these. As you go across the table, you're comparing species X and species Y, the minimum percentage. And so in this case, the minimum is species X. The minimum species X here again, zeros for both, so you get a minimum value of 0. 0 here would be the smaller one, 28 would be the smaller one between these two columns, and we've got a 0, and then 36 is 0, minimum is 0. And then you add up, so we're doing the sum of these minimum values for each, comparing between the two, and 10 plus 10 plus 28 is 48%. Okay, now we're going to do sample 2. So again, we've got these counts. So we've got 3, 4, 9, 8 for the total number of hits here. And then we've got species Y for the total number of hits there, and then the percentage of Y and X. So the percent would be, we're kind of rounding here. Basically it's like the percent out of the total for each of those species. And so we get those values: 12.5%, 16.767, 37.5, and 33.33 for each of those. And so basically, so for species X over here, for example, so 0 out of 24 had that value in there. 4 out of 20 is 1/5 or 20%, so that's how we got those percentages. Well, does that make sense? If it doesn't make sense, email me. But basically, you calculate the count divided by the total gives you your percentage, and then you're trying to minimize the percentage. It's like playing golf. And so here again, we've got these two different values, and so species X is smaller here, so we get 0, 0. Species Y would be the minimums over here. The minimum value for these, they both had values, and the smallest one would be 5, and then zeros again, which leaves us with a sum of the minimum would be 5%. Okay, got through PSI. So now we're going to calculate IRI and PSI for each size group of each species. And so we're going to go ahead and the first example of each is done for us. And so I've already gone through and done these calculations, but I've kind of explained how we're going to go through them. I really recommend that you go through with your calculator and do the math yourself just to double check. These values were all what we got when we went through the lab together in class, but I'm kind of just going to, for the sake of time, fly through them here and there, but the examples have been done for you at the beginning. All right, so first off, we see we've got small bluegill, less than 70 millimeters long. Our sample size is 4 total bluegill. And then we've got these 6 different categories of prey going along the left side here. And so we've got the weight in grams of each of the diet items, and that's like a total weight across the 4 individuals in those samples. And then we've got the number of prey. So we had like 3 chunks of detritus in one stomach total, or across all the stomachs. We've got 77 chironomids, which are just like little midge larvae across all of the stomachs, 0 eggs, and then 1 caddisfly across all the stomachs, for a total of 81 different unique prey items in all 4 stomachs. And then we're calculating a weight percentage. And so basically, like here's the number of stomachs that had those things in it. So 3 of the 4 had detritus. 3 of the 4 had chironomids, 0 had eggs, and 1 had a trichopteran. And so you're going to take your numbers and times your weights, right, to get your percent num, or excuse me, your number times your number to get your percent num. And so that's how we got the 3 times the 0.03, giving us 33% of your total. Oh, excuse me, sorry, wow. Whew, okay, struggling here. So let's go down to the frequency first. So the percent frequency would be, we have 3 fish out of the 4 bluegill, so that's 75% had detritus in them. Same thing with the chironomids, 75%. 1 of the 4 had trichoptera, so that's 25%. The percent number would be out of this total number 81, out of the total value of that, 3 out of 81, 77 out of 81 would be that value of 95. 1 out of 81 would be approximately 1% total. And then out of the weight, so 0.3 out of 0.09 would be about 33% of the weight. 0.05 out of 0.09 is about 56% of the weight. And then 0.01 divided by 0.09 gives us about 11% of the weight. Sorry, I kind of like butchered my way through that at the beginning, but hopefully now as we've gone through that, that makes a little bit more sense. So now that we've figured out how to calculate our percent weight, our percent number, and our percent frequency, we can use those 3 values to calculate our IRI. And so we're taking that percent num plus the percent weight, adding these 2 together and taking it times the frequency. And so 33 plus 4, do those values first, right? And then take that times 75 gives us 2,778. For this one, it gives us 11,296. We get value 0 again for the IRI here, and 309. And so we see the differences. We can say that the chironomids have a high index of relative importance compared to the detritus or the trichoptera. Okay, next we're going to look at large bluegill. And so we're going to go ahead and do some of these together. And so our, let's see here, we've got terrestrial invertebrates at 0.65 grams, we've got Asiatic clams at 0.6, 2.09 for detritus, chironomids, we've got 0.04 grams, 0.01 grams eggs, and no grams of trichoptera. These are the numbers of each of those individuals, or of each of those diet items across all 13 of the large bluegill individuals. These are the number out of the total that had that particular type. And so then if we do the 0.65 divided by the 3.39 again, we get our percent weight. If we do the 5 out of the 48%, we get a percent number, like 10.4 and so on. And then if we do 5, the number 5 out of 13 to get 38% is the percent frequency. 2 out of 13 would be 15% and so on. We get all these values, and then we can go ahead and take 19.2% plus the percent num, which is 10.4%. Going to give us 29.6%, right? And then times the frequency, 38%, gives us, so 29 times the 38% will give us 1,125, and so on. So we see here that detritus has the highest index of relative importance compared to the terrestrial invertebrates. All right, next going down to the small crappie. These numbers are a little bit easier to work with. We've just got 5 small crappies. And so none of them had fish in them. There was 0.3 grams of chironomids, giving us a weight of 10.3% weight. We've got some terrestrial invertebrates here at a total of 82.75% of the weight, and then just a little bit of detritus and zooplankton at 3.4% and 3.4% respectively. 3 out of the 5 had fish in them, giving us 60% frequency. 1 1 out of the 5 had the others, and so that's where we get the 20%. And for the percent nums, 5 out of the 10 total items were Chironomidae. 2 out of the 10 total prey items were terrestrial invertebrates, giving us 20%. 1 out of the 10 total prey items was detritus, giving us 10%, and 2 out of the 10 zooplankton were zooplankton, giving us a percent number of 10%. Okay, let's see here. I think that should have been on 1 originally. I think I might have a typo in there. No, maybe. Okay, so we've done IRIs for all of these. Basically, I'm not going to go through all of these, but I have pulled out some values here that we've calculated together. So you can check off of these to see if you're right. These we calculated together as a class, and so if we did our math wrong, you can tell us about it next week. Okay, and feel free to back up the video and pause if you need to look at those and check your work. Okay, so now we're going to do the proportional similarity index. So remember, S1% is the number of prey item 1 divided by the total number of prey items for that species and that size. And same thing just for S2%. And then it's the sum of the mean column is our PSI. And so basically, it was supposed to have you bring the num columns for each of those size classes, either in sunfish, but just for the crappie back down. So that's where we are getting the num values for this table. So basically, out of our small bluegill, 0 of them had terrestrial invertebrates, 5 of them in the large bluegill did. So using those same numbers from up above, we get an S2 of 10 here versus 0 and 35. We're going to minimize that to 0. 4 and 17, and then would be 4. 95 and 27, and then would be 27. 0 and 10, the minimum would be 0, and then of 1 and 0, the minimum would be 0. And so our low golf score is going to be a grand total of 31 for that PSI. All right, now for the white crappie, comparing the small and large, we had 0 fish in the small, 5 in the large, 5 chironomids in the small, 0 in the large, 2 terrestrial invertebrates in the small, 0 in the large, 1 with detritus in the small, and 1 with detritus in the large, and then 2 zooplankton here, and 0 with the large white crappie, for a total of 10 prey items for the small white crappie and 6 for the large white crappie. And so S2 here is going to be 75%. Remember, we're getting the number of values and everything from the tables above, same fish, and so then we've got an S1 of 50% versus 0, minimized to 0, 20 and 0, 0. Between 10 and 25, the minimum would be 10. We take that value, 0 versus 20, the minimum is 0. For a grand total, your PSI is 10 for your proportional similarity index. And then finally, we do that with green sunfish as well, and you can go through this table, but minimizing between 40 and 50, the min is 40. Between 13 and 33, the min is 13. Between 6.67 and 16.67, the minimum would be 6.67. And then since these are both 0, that would be minimized to 0 for a PSI of 60. Okay, and so here's the end of the assignment that I want you guys to think about. This is the bread and butter of it. What do these values mean, right? And so I want you guys to compare the results of both the methods, the IRIs and the PSIs between species. And so are your results comparable between methods? Just take a moment and look at the values. So if we go back, comparing the small and large, we had PSI of 60 for green sunfish, 10 for white crappie. So that's really good proportional similarity for the small and white or enlarged white crappie, and 31%. Excuse me. So yeah, the proportional similarity index. And so we've got 10, 31, and 60. Versus over here, we've got IRIs for green sunfish of 8,000, 3,200, and 3,500 compared to 8,000 again for detritus, so pretty close. Way off on the terrestrial invertebrates between the small and large, so we see that difference. And then looking at the small and large crappie, we see it's really easy with just within one method for the IRIs to compare between the small ones and the large ones. And so if you compare between, like there's a 0 IRI for the small crappie for fish, but then the large crappie, we have a really high IRI of 18,287. And so that tells us that fish is a very important diet item for the crappie. And so for this answer, I would just say, yes, the results are comparable. In the PSI, some of the rare items kind of get magnified, right? And so you see the difference, the discrepancy between the rare stuff and the more common stuff, and like the really heavy stuff versus the lighter stuff. Okay, next up, question 2. From your results, is there evidence for ontogenetic diet shifts? So change in age, right, over time, diet shifts between the age classes of the 3 species. And so just looking at the shift between IRI is kind of a better way to look at this. When we look at the relative importance of like chironomids is really high in small bluegills, and like detritus and caddisflies, some of the smaller prey, but then the terrestrial invertebrates, the Asian clams, become more important in the large bluegill. There's still some overlap, and so we'll say that that's a moderate evidence for a diet shift, but not really stark. Then going down, next I want to look at green sunfish. And so green sunfish, it's pretty much the same in terms of detritus. A little bit of difference in the terrestrial invertebrates between small and large. The Trichoptera, there's a bit of a difference. And then for the eggs, the chironomids and the eggs, there's a stark difference in those IRIs. So I would say moderate. Using the IRI would suggest that there's a moderate difference in diet between the 2 ages of sunfish. But then looking at the PSI, there's pretty good overlap between the 2. So I'd say, yeah, a moderate difference between the 2. All right, and then looking at the small and large white crappie, here's where it gets really distinct, right? So we've got no fish whatsoever in the small crappie under 150 millimeters total length. Really high importance of chironomids and terrestrial invertebrates, as well as some zooplankton and detritus. But then when we get to large crappie, we see that the overwhelmingly most important item is the fish, and then a little bit of detritus as well. And then if we look at the PSIs, it tells pretty much the same story, how it's got a really low proportional similarity index. So that there's very little overlap. It's like zeros versus a different value. And so the means are 0 here, and it's really low. And so that tells us that there's a really distinct ontogenetic shift between small white crappie and large white crappie. Hopefully that makes sense. So when we're calculating PSI, it's kind of like golf. We want to minimize the score, right? But then when we're comparing the metric, the higher your PSI, the lower your PSI, the less similar, and the higher the PSI, the more proportional similarity there is between 2 samples. Hopefully that makes sense. But yeah, the greater the PSI, the greater the similarity, the overlap between the 2 groups. Yeah, okay. So that's number 2. And then was it proper to include detritus as a prey item based on what you know of the ecology of these 3 species? Why or why not? So the first, this kind of requires a little bit of knowledge about what do these three species eat. So bluegill are really omnivorous, right? So we know that bluegill feed on a wide variety of, um, especially aquatic insects, and snails, and vertebrates, things like that. They don't really eat that much in terms of fish, but they are using a wide variety of zooplankton and different invertebrates, and to some extent some detritus. So we see that detritus does make up a component of the diets of both um, small and large bluegill. Um, for the crappie, we see small crappie have just a little bit of detritus in them. Here's the PSI, but then if we look at, especially the IRI shows this. With the crappie, they're really low in terms of the weight consumed and the percent numb. Um, so they've got pretty low indexes of relative importance. In the grand scheme of things, is 268 compared to 2055 or 3618. Um, and so if you think about it, crappie tend to shift towards passivery later in life, right? And so detritus might not be valid to include detritus because it's a rare diet item, right? And that can skew your sample size, especially in the PSI, towards the rare items. So that creates some error in there because you've included a category that's super duper rare for a fish to ever eat. That's just not what the species goes for. And then finally for green sunfish, if we look at the IRIs, actually they do consume a lot of detritus. It is an important value for an important source of prey for both small and large green sunfish. And if we look at the PSIs between small and large green sunfish, both groups consumed detritus, right? And so there's not a huge difference between those two groups in terms of the proportional similarity. There's high overlap between the two, and they both consume detritus. So it's okay if we include it as a prey category for the green sunfish and the bluegill, but probably not for the white crappie, right? Okay, and that was the last of the questions. So hopefully this gives you just a good overview of the diet analysis. If you have any questions about writing this up, feel free to email me. I'll be happy to help. But if you just fill out those values, go ahead and fill out those values and then email it to me and you'll get full points for the assignment. All right, and with that, I think that's it. So yeah, thanks. Have a good day.